Hello everyone. Today we're exploring the classification of cells and their structural diversity, a topic that sits at the very core of biology and pharmacology. Every drug we prescribe, every infection we treat, and every disease we study starts with understanding the cell. So let's get into it. Overview: 2 fundamental cell types. All life on Earth organizes into one of 2 cell types. Prokaryotic cells, found in bacteria and archaea, are simple, compact, and lack a true nucleus. Eukaryotic cells are far more complex, with a membrane-bound nucleus and a full suite of specialized compartments. That single structural difference, nucleus or no nucleus, has enormous consequences for how we treat disease. Prokaryotic cells. Prokaryotes may be simple, but they're incredibly effective. Their key structures include the cell wall for protection, the nucleoid region where DNA is loosely organized, ribosomes for protein synthesis, and in many species, flagella for movement. No membrane-bound organelles, just a lean, efficient design that has persisted for billions of years. And critically, those ribosomes and cell walls are structurally different enough from ours that we can target them with antibiotics. Eukaryotic cell complexity. Eukaryotic cells are a whole different level of complexity. The nucleus houses and protects the DNA. The mitochondria generate energy as ATP. The endoplasmic reticulum and Golgi apparatus handle protein processing and delivery. And holding it all together is the cytoskeleton, a dynamic internal scaffold that gives the cell its shape and enables movement. This level of organization allows eukaryotic cells to carry out processes that prokaryotes simply cannot. Animal cells. Animal cells have some standout features. They contain centrioles, which organize cell division, and lysosomes, the cell's built-in recycling system, which break down waste and foreign material. Notably, animal cells have no rigid cell wall, which gives them the flexibility to change shape. That's especially important for immune cells that need to squeeze through tissues to reach an infection site. Plant cells. Plant cells tell a different story. They have a rigid cell wall made of cellulose for structural support, chloroplasts that convert sunlight into chemical energy through photosynthesis, and a large central vacuole that stores water and maintains the pressure that keeps plants upright. These 3 features alone explain nearly everything about how plants survive and grow. Fungal and protist cells. Fungal cells are in a category of their own. Their cell walls are built from chitin rather than cellulose, and instead of cholesterol, their membranes contain ergosterol. That ergosterol difference is pharmacologically crucial. It's the exact reason antifungal drugs like azoles and amphotericin B can selectively target fungal cells without harming ours. Protists round out the picture with incredible morphological diversity, from amoebas to algae to ciliates. The cell membrane. Every cell, regardless of type, is wrapped in a phospholipid bilayer, described by the fluid mosaic model. Proteins embedded within this bilayer handle transport, cell signaling, and molecular recognition. What makes this especially interesting clinically is that membrane composition varies across species. Bacterial membranes have no cholesterol at all. Fungal membranes use ergosterol. Those molecular differences make the membrane one of the most targeted structures in all of pharmacology. Specialized cell types in the human body. One of the most remarkable things about eukaryotic cells is their ability to differentiate. From a single type of stem cell, the body produces neurons that fire electrical signals across the nervous system, red blood cells that shed their nucleus entirely just to carry more hemoglobin, muscle cells packed with contractile proteins, and epithelial cells that form tight protective linings throughout our organs. Each of these is structurally optimized, down to the molecular level, for exactly one role. Clinical relevance of cell classification. This is where everything connects for us in pharmacology. Bacterial ribosomes differ structurally from human ribosomes. That's why aminoglycosides can kill bacteria without harming our cells. Fungal ergosterol versus human cholesterol. That's the basis of antifungal selectivity. And when we look at cancer cells, their structural abnormalities, unregulated division, altered surface receptors, dysfunctional mitochondria, are exactly what targeted cancer therapies are designed to exploit. Cell classification isn't abstract science, it's the foundation of rational drug design.